Welcome to the Deep Dive. Our mission, as you know, is to take really complex scientific sources and pull out the critical, high-value insights for you. Today, we are focused on a huge hurdle in personalized medicine, specifically colorectal cancer treatment.
That's right. We're looking at colorectal cancer, or CRC, and this persistent problem of resistance to targeted therapy. For years, the KRAS oncogene, which is found in up to half of all CRC cases, was thought to be completely impossible to target.
But the science here has just been moving so fast. We've seen these new drugs like sotorasib and adagrasib, the new generation of KRAS-G12C inhibitors. And when you combine those with EGFR antibodies, cetuximab is a common one, the initial results were fantastic. We're talking objective response rates of 34 to 46%.
It was a major win, a huge success story.
One that hit a wall pretty fast.
And that's really the punchline of our sources today. Despite that incredible initial success, the patient responses are often, well, they're short-lived. The tumors find a way to survive, and patients relapse, usually just within a few months.
Okay, so the cells are fighting back. But how? We know some of it is classic genetic change, right? The tumor gets a new mutation to get around the drug. But what's really fascinating here, and what the sources dive into, is that about a third of these relapsed tumors, they don't have any new detectable genomic alterations.
Which points you directly to a non-genetic escape plan. And the core discovery here is it's pretty dramatic. Colorectal tumors don't just mutate, they change their entire identity. They use something called lineage plasticity to become a highly resistant cell type.
A Paneth-like cell state.
Exactly, the Paneth-like cell state.
So our mission for this deep dive is to trace that molecular switch. We want to understand how this resistant state emerges, what's driving it, and most importantly, how we can block it.
And potentially make these targeted treatments durable for patients.
Okay, let's unpack that in our first segment. This Paneth-like cell state, Paneth cells. I'm guessing that's a term, that's a footnote for most people from a biology class. What are they normally, and why on earth is this small intestine cell type showing up in a colorectal tumor? It's a great question. So Paneth cells are highly specialized secretory cells. In a healthy person, you really only find them at the base of the crypts, in the small intestine. They're sort of the guardians of that little neighborhood. They secrete these potent antimicrobial peptides, defensins, lysozyme, that keep bad bacteria out.
Okay, so there's security guards.
Right. And they also support the LGR5 positive intestinal stem cells, which helps the tissue regenerate. They maintain homeostasis.
So the fact that a large intestinal tumor is suddenly taking on this identity, that's a radical shift. It's like a submarine deciding it needs wings to survive.
That's a perfect analogy. It is purely an adaptive strategy to survive extreme stress. The CRC cells don't become perfect normal Paneth cells. They just acquire the Paneth-like genetic signature. They flip on the genes that give them that survival advantage.
And the researchers used some really sophisticated models to prove this was happening. Walk us through that initial evidence. They started with a model called ICAP. It's a genetically engineered mouse model that's programmed to get really aggressive CRC. It has 3 key mutations: KRAS, G12D, APRIC, and P53.
So it's a very good mimic of advanced human cancer.
A very good mimic. They treated these mice for 2 weeks with a dual therapy, a KRAS G12D inhibitor called MRTX10133, plus that EGFR antibody, cetuximab.
And the therapy worked at first. It suppressed the growth.
Dramatically. But the key was in the cells that survived, the residual tumor. They did a transcriptomic analysis. Basically, they looked at all the RNA in those surviving cells and found this massive, undeniable enrichment for Paneth cell pathways, especially defensins.
And they could see it, too.
They could see it. Immunofluorescence staining confirmed it. The surviving cells were just glowing positive for the Paneth markers, DA5 and LYZ.
So was this just a weird quirk of the KRAS G12D mutation, or is this a universal escape route?
It's beautifully conserved. That's what's so compelling. They immediately tested other models, patient-derived cell lines, organoids, with the KRAS G12C mutation. They used the clinically approved combo, sotorasib plus cetuximab, and saw the exact same thing, Paneth-like cell enrichment.
So this is a general strategy. It's the tumor's go-to adaptive response to this specific therapeutic pressure.
Exactly, regardless of the specific KRAS flavor.
And the sources mention that the cells don't just jump into this state. There's an intermediate phase, this DTP state. What is that?
That's a crucial detail that came out of their single-cell RNA sequencing that could actually map the journey over time. The cells first enter what's called a diapause-like drug-tolerant persister state, a DTP state. Think of it like a deep cellular hibernation.
A holding pattern.
Yes, a holding pattern. The cells just pause, they stop proliferating, they weather that initial therapeutic shock.
So the DTP state is the emergency brake, and the Paneth-like state is the long-term escape vehicle they build while they're stopped.
That's it exactly. The DTP intermediate buys the cell time to rewire itself, find that escape route, and then transition into the stable, resistant, Paneth-like state.
Which brings us to our second segment. Proving this is really plasticity. Are we just selecting for a few rare Paneth-like cells that were already there, or are the main tumor cells actually changing their identity?
The lineage tracing experiments answer this definitively. It is transdifferentiation, a true identity switch. They engineered the cells with this really clever molecular reporter system. So the promoter for DFAT5, that Paneth-specific gene, was set up to drive a color switch.
So before the switch, the cells are one color, and after, they're another.
Precisely. They started out glowing red, but if a cell turned on that DFAT5 promoter and became Paneth-like, it would instantly switch to glowing green.
That is an amazing way to track it in real time. What were the numbers?
The numbers of the smoking gun. Before treatment, the green Paneth-like cells were almost nothing, 0.23%.
Okay.
After just 2 weeks of the dual therapy, that fraction skyrocketed to 32.26%.
Wow, from basically 0 to almost a third of the surviving population.
That kind of massive induction can only happen through a lineage switch. It's definitive proof.
And what's really telling about the adaptability of cancer is that the state isn't permanent.
That's right. The sources show it's completely reversible. When he took the drugs away, the Paneth-like population just plummeted.
Which is so important because it means we're dealing with an adaptive survival program, not a new, stable genetic mutation that's been locked in.
The cells are only doing this because the drug is forcing them to.
So once they have this Paneth-like identity, how does it actually protect them? What's the biological superpower it gives them?
The shield it provides is enhanced MAPK pathway reactivation.
Okay, let's pause on that. MAPK reactivation. For everyone listening, the whole point of these drugs is to shut down that pathway. It's the cell's accelerator pedal for growth. So is the Paneth-like switch just a secret way for the cell to get its foot back on the gas?
That is the perfect analogy. The goal of the therapy is to suppress the PERK, to shut off that signal. But the Paneth-like cells found a way to maintain higher levels of KRAS-GTP, which is upstream, and they showed a much stronger rebound of the MAPK signal after treatment. Staining confirmed it. The puric levels, the actual engine activity, were way higher, specifically in the DFAT5-positive Paneth-like cells.
They changed their identity to reactivate the very pathway the drugs were designed to block.
Yes.
So we've established the what MAPK reactivation and the how trans differentiation. But this leads to the biggest question. What's the master switch? What single molecular driver is forcing the cell to do this?
To find that, you need some serious power tools. And in segment 3, that's what they use, an unbiased CRISPR knockout screen.
This is like systematically turning off every light switch in a giant factory to see which one shuts down the whole production line.
A great way to put it. They targeted 1,741 human transcription factors, the master gene regulators, in 3D tumor spheroids. They were looking for any transcription factor where losing it suddenly made the cells super sensitive to the drug combo. If losing it makes the cells die, it must be essential for resistance.
And the toplet was a name that's actually pretty well known in gut biology, SMAD1.
Exactly. SMAD1 was the undeniable top hit, and it made immediate sense. SMAD1 is part of the BMP pathway, which is critical for regulating cell identity in the intestine.
It's a known player in differentiation.
It is, and the proof was there. First, the dual therapy itself caused SMAD1 levels to go way up. And then, crucially, using microscopy, they saw that the SMAD1 protein was co-localizing. It was right there in the same spot as the Paneth marker DFAT5. It was turning on exactly where the identity change was happening.
So what happened when they actually knocked it out in a living tumor?
The results were just dramatic. Knocking out SMAD1 made the CRC spheroids incredibly sensitive to the drugs. And in the xenograft models, the mice with human tumors losing SMAD1 restored sensitivity. They saw major tumor regression in 70 to 90% of the mice.
And it blocked the identity switch.
It almost completely eliminated the Paneth-like state transition. It showed that SMAD1 is essential for this entire escape route.
Now, a really key detail you mentioned is that this activation is non-canonical. For most people, SMAD1 works with a partner, SMAD4. Why does it matter that SMAD4 wasn't involved here?
That's a huge tell. It means this isn't a normal physiological response. It's a stress-induced, hijacked pathway. though normal targets of the SM81 pathway were actually downregulated, and its usual partner, SM84, was totally dispensable for resistance. So the therapeutic stress basically hijacked SMA1, forced it to go rogue, and gave it this new non-traditional resistance function.
So if SMA1 is the rogue driver, what is it turning on downstream that actually causes the resistance?
The pathway analysis pointed right to it. The genes most correlated with SMAD were all tied to FGFR3 signaling.
Fibroblast growth factor receptor 3.
Yes. And FGFR3 was one of the top genes upregulated after the dual therapy stress.
And they could prove SMAD was directly flipping that switch.
They did. Using techniques like CHOP-EQ-PCR, they confirmed that SMAD physically binds to the promoter region of the FGFR3 gene and actively drives its expression.
So there's the full axis. KRAS-EGFR inhibition leads to non-canonical SM81 activation, which then turns on FGFR3.
That's the chain of command.
And this is where the therapeutic opportunity really opens up. Our fourth segment. Because FGFR3 is a known targetable receptor, if that's the functional link driving the Paneth-like state, then blocking it should, in theory, solve the whole problem.
It absolutely should, and the data shows it does. FGFR3 is the druggable vulnerability in this whole story. They use pan-FGFR inhibitors, drugs like sudabatinib and infigratinib, and these are important because they are already FDA-approved drugs for other cancers. They're clinically relevant right now.
So you add a third drug. You create a triple therapy, the original dual treatment plus an FGFR inhibitor, and that restores the tumor sensitivity.
It's the powerful conclusion of this work. In their mouse models, the triple combination KRAS inhibitor, EGFR inhibitor, plus futibatinib was just vastly superior. Futibatinib by itself did almost nothing, which is key. It shows its only job here is to block that specific escape route.
And what was the outcome? Did it just slow things down?
It did much more than that. It induced profound and sustained tumor regression. And here's maybe the most incredible finding. In some of the ICAP models, the triple therapy was so effective it caused a complete reversion of the tumor back to a benign adenoma.
They demalignify the tumor by blocking its non-genetic escape plan.
That's essentially what they did.
And crucially for all of this, they confirmed it happens in actual human patients.
Yes, that's the final piece. They analyzed paired biopsies from 2 CRC patients on KRAS-EGFR inhibitor therapy. In both cases, the residual tumor tissue they collected after treatment showed that same marked enrichment of DFAF5-positive Paneth-like cells.
This isn't just a lab curiosity. It is the active clinical resistance mechanism.
It is.
So to bring this entire journey full circle for you, we've traced the whole mechanism. The therapy stress kicks off a temporary DTP state. This leads to non-canonical SMAD1 activation. SMAD1 then turns on FGFR3, which forces the tumor into Paneth-like plasticity. And that new identity allows for MAPK reactivation and ultimately treatment failure.
And the real knowledge gain is that we can make the original dual therapy durable again by simply adding an existing third drug and FGFR inhibitor to block that plasticity before permanent resistance develops.
This really is a compelling case. It suggests that clinical trials combining FGFR3 inhibitors with this dual therapy need to be a top priority.
I think so. And as you're processing all this, here's a final provocative thought for you to consider. We learned that this Paneth-like switch is specific to colorectal cancer. It's different from the plasticity we see in lung or pancreatic cancer. So, if the tissue of origin dictates this specific escape route, how might future cancer diagnostics evolve to predict a tumor's escape plan before treatment even starts?
Could we identify markers that flag a tumor as being susceptible to a certain kind of plasticity and then block it from day one?
That's the question. How do we get ahead of the tumor's adaptability?
A fantastic question for future research. Knowing the enemy's escape plan is half the battle. Keep digging into lineage plasticity, and we'll see you on the next deep dive.